In the last class, we were studying genetics and talking about how genetics is the study of inheritance. And we talked about the work of Gregor Mendel and how he learned that there was this passing on of information from parent to offspring. He learned that the parents couldn't pass on information that they didn't have. So if a parent was true breeding for a trait, they couldn't pass on a different characteristic for that trait. And so he realized that there had to be something physical that was being passed down from the parents to the offspring in their gametes. And that was a really, really cool and interesting discovery. But the coolest thing is that he was able to make some predictions about the outcomes of the offspring based on this genetic information that he had. So in a previous lecture, we talked about we've got a heterozygous individual. We have him right here. And if you cross that with a homozygous recessive individual, then you can track what their offspring would look like. And you can make some predictions. You could say, well, I'm going to have 50% that are going to show the recessive trait and 50% that are going to show the dominant trait. And you can even say, oh, and the 50% that show the dominant trait, they're heterozygous. So we practiced all of that vocabulary last lecture, but now we're going to take it to the next level. I know that you know I am a fan of Mr. Anderson and his work. I think he does a great job of explaining things. Don't you, Mr. Anderson? So, today we're going to have him help explain dihybrid crosses, which are between two different traits.
Usually students do fairly well on those. Next are dihybrid crosses, and this is where the mistake really starts. And so if we look at this parent, this is a typical dihybrid cross. Let me tell you what the letters stand for. The R stands for round pea seeds, and the Y stands for yellow seeds. If it's the recessive, that stands for wrinkled, and if it's a little y, that stands for green. And so we're looking at a dihybrid, so that means two traits. We're looking at seed shape, round or wrinkled, and seed color, yellow or green. So now we have to do a dihybrid cross. And so the tendency, if we look at this parent, the tendency is to see that there are 4 letters over here, there's 4 boxes over here, and then you just simply write them out. Big R, little Y, little y, and then you get a weird answer and you don't know what to do with it, okay? That's wrong. That's a mistake, okay? Whenever you're figuring out the gametes, remember that you have to give one of each letter. In other words, each of the gametes is going to have one of each of the alleles. And so let me clear this mess out of the way. So what do? Let's say this parent right here. And I'm going to write it up here so it makes a little more sense. Big R, big Y, little y. What possibilities could they produce? By giving one of each letter, remember. Well, they could give the big R and the big Y. So big R, big Y. That'd be one possibility. They could also give the big R and the little y. So big R, little y. They could also give the little r, big Y, or the little r, little y. Since they're giving one of each color, there's only 4, one of each letter, there's only 4 possibilities that they can give. So it would be kind of a mess, but those are the 4 right here. And so those 4 are going to go across the top. So we've got big R, big Y, little y, little r, big Y, little r, little y. So those are the 4 gametes that you could produce. In other words, with this parent, you can only get 4 combinations of each of the 2 letters. Same thing down this side. So it's going to be same thing on written down this side because this other parent is going to be exactly the same. Okay, so it would take me a long time to write all of those out. So let me throw those in here. So these would be the parents, all the possible gametes you could get. And if I fill those in, these first 9, if you look at them, let me get a color. That's different. Let me grab a yellow color. And so this one right here is going to be round, and it's going to be yellow. So it's going to be round and yellow. And this one you can see, it's going to be round and yellow, round and yellow, round and yellow, round and yellow, and round and yellow, and round and yellow, and round and yellow, and round and yellow. In other words, 9 of them are going to be round and yellow in shape. And the only reason why is that the R is dominant. And so you could have one like this where they're hybrid for both, and they're still going to be round and yellow. And so let me try to add the next ones. So what about these next ones? Well, the next ones are going to be round and green, so we'll get a different color. So these ones are going to be round and green because the round is dominant, but they don't have the dominant for the yellow, so they're not going to be. Get rid of that. Let's have the next ones. So these ones are going to be wrinkled and yellow. So again, I got to get a different pen. So these ones are going to be wrinkled and yellow. And then if we look at the last one, it's going to get all of the recessive alleles. And so that one's going to be green. It's going to be green and wrinkled. Okay. And so when we say there's a 9 to 3 to 3 to 1 ratio, that's just a typical dihybrid cross. We're going to have 9 of the phenotypes that are round and yellow, 3 of the phenotypes that are round and green, 3 of the phenotypes that are yellow and wrinkled, and then only 1 of the ones that's not. And that's only going to work if you are able to set up your gametes correctly on the side. Now, it's super hard for you to answer a question like this on a test. You're rarely going to have to draw a dihybrid cross. But it's important that you understand the concept of it, because most of the genes inside your body are not caused by one gene, they're caused by multiple genes. So how tall you are is caused by probably a dozen different genes inside your body. So you can imagine how big the Punnett square is going to be for that.
Like Mr. Anderson, at this stage in the game, I don't require you to learn how to do a dihybrid cross, but I certainly will help you if you decide that you would like to know, because it's something that you'll need to know as you go into AP Biology. However, in this class, you will have to fill out or read a dihybrid cross. So, let's go over how to do that. You can see that here we've got, oh, I lost my mouse, there we go. We've got big P, big S, big P, little s, little p, big S, little p, little s. And then over here we have the same. We have all of the possible connections. Big P, big S, big P, little s, then little p, big S, little p, little s. So that's how we get all of the possible combinations that you can make. You can actually kind of do this whole FOIL thing. So remember in algebra, it was like first, outside, inside, last. If that helps you remember, you can consider these first, outside, inside, last. You consider these two separate units, and then you can do FOIL on it. Whatever makes you happy. But anyways, like I said, you're not going to have to set them up yet, so don't panic. I do, however, want you to know that when these two come together, you're going to get big P, big S, big S. Big P, big S, little s. Big P, big S, big S. Big P, big S, little s. So I'd like you to go ahead and finish this, and I'd like you to write down the phenotypes and the ratios below. Big P is purple and little p is white. Big P is dominant. Smooth is dominant to wrinkled. Smooth is represented by big S and wrinkled is represented by little s. Go ahead and pause this while you take the time to try to set this up and then work it out. Alright, so let's take a look here. You should have gotten all the rest of these, big P, big S, little s, and finish these all out. And you should have ended up with some that were purple and smooth, some that were purple and wrinkled, some that were white and smooth, and some that were white and wrinkled. When you count them up, you should have ended up with a ratio of 9 purple smooth to 3 purple wrinkled to 3 white smooth to 1 white wrinkled. So it should be a 9:3 to 3:1 ratio. And that's a pretty common ratio to look for in any test that you take on a dihybrid cross. All right, kiddos. So I got to admit, sometimes things don't always work out the way Gregor-Mendel wanted them to. There are all sorts of things that go differently than just his perfect little scenario with those perfect little pea plants and only 2 different alleles. So remember we talked about like how eye color can have blue and brown and green and purple? Well, Gregor-Mendel didn't talk about having 4 different alleles, right? That complicates things. That's called multiple alleles. Okay, so if you have lots of different alleles for one trait, that's called multiple alleles. Polygenic traits have several different genes that code for one trait. So it's not just alleles. For instance, your skin. Your skin is coded for in a whole bunch of different genes. And so it does things like how much yellow pigment you have in your skin and how much brown. And so that's going to determine whether you're olive-skinned or not. So there's different colors to the pigments, and so there's 6 different genes that code for your skin color. Right? That's why we have such a really cool variety of skin colors on our planet. There's so many genes that are playing a role. That's called a polygenic trait. One of the cooler ones, though, that we're going to talk about is codominance and incomplete dominance. Those you do need to know how to do a Punnett square for. So let's take a look at this scenario over here on the right. We have a red flower. It's mixing with a white flower. All of a sudden, we get pink flowers. That is not according to the law of dominance. It should be red is dominant to white, red wins. But that's not what happened. They compromised. So, when that's the case, you use 2 capital letters, and they're different to represent each different allele. And so R and W together, we know that red and white together make pink. And so we put big R on the top, big W on the side, and then the ones that are RW are going to be pink. Okay? So that is incomplete dominance. So they're both compromising. An incomplete dominance, they're compromising. But in codominance, it's a slightly different story. In codominance, it's not a compromise like it is in incomplete dominance. Instead, in codominance, you have both genes equally represented. So check this out. You've got one red parent, there you go, up here, and you've got the white parent over here. But R and W doesn't make pink, it makes party colors, right? So you've got both red and white that are demonstrated. So let's compare. Incomplete dominance, let's think cow, white, other cow, black. Incomplete dominance, the babies are gray. Cow, white, cow, black. Codominance, the babies are black and white. Okay? So, let's play again. Let's say, I have a bunny rabbit, and it is, let's say, brown, and I cross it with a black bunny rabbit, and I get a bunny rabbit that looks kind of brindley. It's a mixture of black and brown. What would that be? Right, codominance. Let's say I have a cat that's gray and a cat that's white, and I end up with a gray cat with a white face and white feet. Again, codominance. It's only one or the other, gray or white, and they're both showing up. But let's say I have a black cat and a gray cat, and I get a dark gray cat. That's incomplete dominance. Good. You're doing pretty well. Here's a look at blood typing. You can see that 37% of people in the U.S. population are a type of O blood type, and 34% are type A positive. We're going to spend more time in class discussing blood type, but I kind of wanted to give you a quick general overview. It's done a little bit differently because there's 2 dominants and 1 recessive. So if you were to take a look, I lost my mouse again. You guys can probably see it, but I can't. Okay, so you can see this is A and B are both dominant. Okay, they're co-dominant with each other. Okay, but little I is the O blood type, and it is recessive. So if I had A with this one, so if this was my allele, A and little I would have the A blood type. If I was homozygous for big A, I would have the A blood type. If I was homozygous for the dominant big B, I would have the B blood type. And even if I would had B and O, like the little recessive, I would have the B blood type. But if I have A and B, that's where I get the AB blood type. Little I is the only way that you can get O. But it must be fairly common for 37% of the population to have the O blood type. In this class, we're not going to go over the positive and the negative. That's called the RH factor. We might bring it up a little bit when we do our lab activity, but you're not responsible for that at all. Remember when I told you that polygenic traits are pretty funky? So this right here just shows you the 3 traits or 3 genes that can code for the different levels of pigment. So this is really pale. This is when you get the pale allele from each of the 3 genes, and each one of these represents 1 pair of alleles. So this is from mom, this is from dad. So mom gave you 3 pale, and dad gave you 3 pale. So you're going to be really pale. You're going to be down here in the bottom of this part. Okay, but as you go up, you're getting more and more dark color skin genes until you get to the most dark or the most amount of pigment can be in your skin, right? Okay, that's complicated, but understandable. But now look over here. So these are some of the other colors that can be integrated into your skin. And so it gets a lot more complicated than that. So, like I said, I just want you to know that polygenic traits exist, that they are traits that have lots of genes that code for the same characteristic that we're looking at. So if you were looking at skin color, we'd be looking at not just these 3 genes, but the genes that code for these pigments as well, which is pretty insane. So does the environment have a role in how genes determine traits? Well, here's the deal. It totally does. If your parents were really, really super cruel, and when you were first born, they locked you away in a closet, you probably wouldn't look as gorgeous and tall as you look today, right? No, you'd be stunted, and you wouldn't develop correctly in your brain, your body wouldn't develop correctly. So environment does have something to do with genetic expression. So if you look down here on the left-hand side, you can see there's a guide to nutrient deficiency symptoms. But what you can see is that some genes are actually turned on by the different nutrients that you have. So in these cases, you can see that different colors or different characteristics were turned on by either the presence or the lack of specific nutrients. And so that's led to different phenotypes. And so we say that the environment can have a role in how your genes are expressed, and the expression of genes is going to give you your different phenotypes. Here and here you can see that there's some negative outcomes, and the plant just doesn't look healthy, and so it looks like it's probably struggling a little bit because it doesn't have enough of its nutrients, right? But in this one, you can see that that's just caused a different color variation, which is kind of interesting and a cool idea. In this really, really cool experiment, The scientist was looking at these different Himalayan bunnies and Siamese kittens and said, why do they have just black on their ears, their nose, and on their paws and tails? And she was like, those are probably the coldest parts of the body. And if they were black and the sun hit them, then that would help warm those parts of the body up. And so she made this hypothesis that it wasn't just like a genetic pattern, that instead it was dictated by temperature. And so what she did, she shaved off a little patch on the bunny rabbit, and then she put, the patch would have been like right here. Then she put an ice pack on the bunny rabbit for a little while, not enough to hurt the bunny. The bunny was fine. But then when the hair grew back, it grew black. And that supported the idea that the temperature of the environment actually changed the expression on the gene on those other locations where it's naturally dark. So that was another cool experiment that showed that environment changes the expression of genes. So what are the take-homes? The first take-home is how to fill out a dihybrid cross and figure out the different phenotypes and the different ratios of those phenotypes. The second is that Gregor Mendel made some great observations about traits, but he picked traits that only had 2 different alleles for the trait, or 2 different variants for the same trait, and were easily observable. But there's a lot more than that going on, and so we started talking about funky genetics that don't behave the way that Gregor Mendel said that they should. We talked about this idea of incomplete dominance when you make a compromise, co-dominance when you get a party outcome, right? So you end up with this party-colored flower, and it shows both characteristics. We talked about how sometimes there can be multiple alleles for the same trait, like in blood typing or in eye color. So in blood typing, we talked about there were some cool things going on. A and B were both dominant, and type O was recessive, and there was all sorts of interactions that could happen there. We talked about polygenic traits, when you have lots of genes coding for the same trait, and how that can change the expression of a trait, or can change the way that we take a look at genetics. And then we talked about even the environment can change the way that a gene is expressed in an individual, not just in plants, but in animals. And so that kind of wraps up our day where we're talking about funky genetics. And I hope you had fun with me today, and I'll talk to you real soon.